let us see the mechanism of the drug action. So if a drug after entering into the body, how it is showing the action. So there should be some mechanism how the drug shows its effect. So if we see, before going into the exact topic, so here once the dose is given or a drug is given into the body, from the tablet or from the preparation or from formulation, the drug has to be released and it has to reach the blood circulation. So that part is called as pharmacokinetics. So the pharmacokinetics is a process from where the drug is released and it is reaching to the systemic circulation. And here once the drug is released into the blood, it has to bind to some site. So it has to bind either to the receptor or it has to bind to some enzyme. So it is binding and thereby it is exerting its effect. So this process is called as pharmacodynamic process. So here the drug, after it is there, after it is released, it has two things to do. So one is that it binds to the plasma proteins and the other one is it is in the unbound form or free state. So whatever the drug after absorption it is there, the free drug which is there, it either binds to the plasma proteins or it undergoes metabolism. Few portion of the free drug undergoes metabolism, or few portion is eliminated out of the body, or whatever the remaining drug, the drug which has to show its effect, it will go and bind to the receptor. It will either bind to the receptor or it will either bind to the enzyme to show its effect. So let us see what are the target sites where the drug is binding. So the drug mainly binds to either receptors, or it binds to the ion channels, or it binds to enzymes, or it binds to the carrier molecules. So based on the mechanism or based on the site of target, or based on the target where it is binding, so the mechanism of drug action, it is divided into the receptor mediated action and non-receptor mediated action. So receptor mediated action, in the sense the drug is binding to the receptor and it's showing its action, whereas non-receptor mediated in the sense it binds to either ion channels or carrier molecules, or it binds to the enzymes in order to show its action. If we see a receptor. So what is a receptor? The receptor is any cellular macromolecule that a drug binds to initiate its action. So it is a macromolecule, mostly it is in the form of protein, and where it is a place where the drug binds and the drug shows its effect. And what is a drug? So a drug is a chemical compound which, when binds to the receptor, show its biological effect. In the sense it brings some alterations in the physiological as well as biological effect of the body. And here all drugs may be chemicals, but all chemicals are not drugs. So all chemicals won't alter the biological effect. So they may not show the positive effect to the body. So all drugs may be chemicals, but all chemicals are not drugs. And if we see the drug, how the drug is acting, so it is acting by a mechanism of enzyme inhibition. So what is this enzyme inhibition? So to an enzyme, a substrate binds and thereby the enzyme shows its effect called chemical reaction. Here, if we see the enzyme inhibition, here the inhibitor which is there, or the drug which is there, it will not allow the substrate to bind to the enzyme. The inhibitor will bind to the enzyme, thereby this inhibitor is inhibiting the action of enzyme. It is not allowing the enzyme to show its action. So this is one type of mechanism by which drug act. The other is drug receptor interaction. So here the drug is binding to the receptor and it is interacting with the receptor, and thereby it is showing various effects. Non-specific interactions are those where the actions are exhibited by the ion channels. The drug will bind to the ion channels and indirectly it is altering the body functions. And dose response curve. So dose response curve is a curve, or else it is a graph where it is plotted against the activity and concentration. So here, as the dose increases, the potency or the activity of the drug is increasing. So here we can see two graphs where as the dose is going on increasing, even the efficacy or potency of the drug is increasing as such. And the receptor mediated action. So let us see what are the various receptor mediated actions, because these are the main mechanism of action by which the drug is acting. So we saw a receptor is a protein substance or a macromolecule where it is having some space for the drug to bind to it, and thereby the drug shows its effect. Here if we see, the drug will bind to the receptor and thereby it is showing response. If we see lock and key model, so the drug and receptor are like lock and key model. So the lock which is there, it is having exactly the shape of the key, and thereby the key fits into the lock and thereby the lock is getting opened. So in the same way, the drug is key here and the receptor is lock here. So the drug called, which is look like key, it is binding, it is having the same structure exactly which is on the receptor, and it is exactly fitting onto the receptor, and thereby it is showing the action. Here the drug which is binding to the receptor, it is having the various reasons, or else it is having some various mechanisms. For example, how it is binding. So why the drug is binding to the receptor? So one thing is that it is having affinity. Affinity in the sense the capacity of the drug to get attracted to the receptor. Like key. Key is having the affinity to lock. Then only it is getting removed. So in the same way, the drug, it is having the affinity. The receptor is attracting the drug towards it, and thereby it is forming the complex. And intrinsic activity or efficacy, in the sense, it is the ability of the drug to trigger the pharmacological response after making the drug receptor complex. So once the drug, if it is binding to the receptor, the drug is having the ability to alter the pharmacological effect. The body functions are getting altered. So this is called intrinsic activity or efficacy of the drug. And ligand. So ligand is a compound or any chemical substance which is binding to the receptor at its site. And so what are the types of drugs? So the drug may be of various types, like agonist, which when bind to the receptor show the pharmacological effect. Antagonist is a compound or drug which when bind to the receptor will antagonize or will reverse the effect of the other drug. And partial agonists are those which potentiate slightly the action of the agonist. And inverse agonists are those molecules which completely antagonize the action of the agonist. Here if we see the graph, see the drug molecule or neutral antagonist is the one which is not having any effect. And inverse agonist is a compound which will reduce the efficacy of the drug molecule. And partial agonist is a molecule which slightly increases the agonist action. And fully agonist is a compound which is completely increasing, or else which is completely showing synergistic effect. And if we see this diagram, normally before any drug binds, the receptor is receptor is free, and there will be normal cellular activity. But here when agonist drug, so when an agonist it comes and bind to the receptor, it is showing more action when compared to the normal cellular activity. Here we can see the cellular activity is enhanced. And if we see the antagonist, when the antagonist comes and binds on the receptor, then there will be no cellular activity, in the sense the cellular activity is blocked by the antagonist. Now here the drug which is binding to the receptor, so it should be have some bonding. So the bonding where the drug is binding to the receptor, the bond by which it is forming is of various types. So one is hydrogen bonding, the other one is ionic bonding, the other one is van der Waals forces, and the other one is covalent bonding or coordinate covalent bonding. So by these various bonds, the drug is binding to the receptor. Now let us see what are the types of receptors which are present in the body. So there are 4 types of receptors. Type 1 is inotropic receptors or ion channel, ligand-gated ion channel receptors. Type 2 is metabotropic receptors or GPCR receptors, that is the G protein coupled receptors. So these GPCR receptors act by the 3 different mechanisms like adenylyl cyclase pathway, IP3, that is inositol triphosphate or DAG, diacylglycerol pathway, and ion channel regulation. The other type of receptors are enzyme linked receptors, which is following 2 pathway mechanisms. One is intrinsic enzyme receptors and the other one is JAK-STAT kinase pathway. JAK-STAT in the sense Janus kinase and STAT kinase pathway. The type 4 receptors are receptors regulating gene expression, which are also called as nuclear receptors. Now let us see the ligand-gated ion channel receptors. So here this ligand-gated ion channel receptors, these are also called as cell surface receptors. So these receptors, they are present on the cell surface and thereby they will allow the drug to bind at their site. So mostly these drugs, they act by the ionic mechanism, in the sense when a drug binds to these receptors, they will either increase or decrease, or else they maintain the concentration. They will alter the concentration of ions like sodium, potassium, calcium and chlorine, which are present in the body. And agonist, when binds to the receptors, it will open, or else it will open the receptor and thereby it will allow the ions to get enter it. And the entry or exit of ions is based on concentration gradient, in the sense if the concentration is more inside the receptors, then the ions will move inside to out. Whereas if the concentration is high outside the receptors, then the drug molecules move outside to inside of the receptor. So based on the concentration gradient, the movement of the ions is done. If we see the examples of ligand-gated ion channels, they are nicotinic cholinergic receptors, GABA A receptors, glycine receptors. These are inhibitory receptors. They show inhibitory actions. And excitatory amino acid receptors like kinase receptors, NMDA receptors, as well as quisqualate receptors, and 5-HT3 receptors, that is serotonin receptors. And if we see here the GABA receptor, so GABA receptor is having a 5 unit. So here the ion channels all have 5 subunits, 2 alpha units, 2 beta units and 1 gamma unit. So when this GABA, that is a neurotransmitter or drug, it is binding to its site, then the receptor will open, it will allow the chloride to get enter into it and thereby it shows the effect. The other type are NMDA receptors. So here the glycine or glutamate receptors, these are inhibitory type of receptors. So these receptors are having 2 sites. It is having the binding site for glutamate and it is having the binding site for glycine also. So the glycine will bind to glycine binding site and glutamate will bind to glutamate binding site, and thereby the channel, the receptor gets opened and the calcium is entered into the receptor and thereby they are showing the effect. Now, if we see the features of these receptors, these are the protein molecules which form the water-filled pores. So here the receptors are protein in nature. So when water is filled, so they form a pore. When the fluid is filled into them, they form pores. And thereby they are getting opened, and through these pores the ions are getting entered into the channel, and this entry is based on the concentration gradient as well as the state of the receptor. So if the receptor is in opened state, then only the ions can get entered. If the receptor is in closed state, it won't allow the ion channels or its ions to get entered into the receptor. And if we see the molecular structure, it is ligand gated. So it is having a space for ligand. So it is having a space for ligand to bind to it, and it has the 4 subunits, in the sense 2 alpha are there, 2 beta are there, and 1 gamma and 1 delta are also there. And if we see it is having a few compounds or few receptors are having pentameric structure, in the sense they are having 5 subunits in them, and each subunit spans the membrane 4 times. So each subunit span is 4 times. If a drug binds 4 times, then the span of that subunit is over. And if we see how the ions are affecting, so if the sodium and the calcium levels are increased, then it show excitatory effect, and if these levels are decreased, then it show inhibitory effect. And if potassium levels are increased, it show inhibitory effect, and if potassium levels are decreased, it show excitatory effect. And if chloride levels are increased, it shows inhibitory effect, and if the chloride levels are decreased, then it show excitatory effect. So based on the concentration of the ion also, the action, whether it is showing inhibition action or excitatory action, it is based on the receptor. If you see the importance of these receptors, so these receptors, they mainly aid in the generation, propagation of nerve impulse. So if a drug is binding to these receptors, they aid in the generating the nerve impulses, and they aid in the synaptic transmission of neurons. So the neurotransmitter, when it is released in the synapse, it is binding to these receptors and it is showing the action. So this transmission, signal transduction, is based on these receptors. Muscle contraction, salt balance, the ionic concentration balance, hormonal release, as well as muscle relaxation, anti-arrhythmia, and anesthetic action. So all these actions which are there, these are mediated by the ion channel activation or inhibition. The other type of receptors are G protein coupled receptors, which are also called GPCR receptors. So when compared to all the four receptors, these are the largest family receptors. So they are having high number of families in them, and they are having a special molecule called G protein, which is aiding in the mechanism of this receptor. So when agonist, it is binding, the agonist binding site which is there, it will face the extracellular. So this is the membrane, this is extracellular space and intercellular space. The agonist binding site which is there, it is facing the extracellular space. So the agonist will come and bind here in this way, and the recognition site formed by the cytosolic segments will couple with the G protein. So here whatever the recognition sites are there for binding for the drug, they combine or they coordinate with the G protein in order to exert their mechanism. If we see the examples of GPCR receptors, muscarinic cholinergic receptors, GABA B receptors, dopamine D2 receptors, beta adrenergic, alpha 1 adrenergic, alpha 2 adrenergic, and few 5-HT receptors. If we see the molecular structure of this GPCR, it is having 7 helical structures. In the sense, it is having 7 cylindrical structures which are called helices, and these 7 structures are connected with loops. So 3 loops are present extracellularly and 3 loops are present intracellularly. If we see, this is the structure, it is having 5 helicals, H1, H2, H3, H4, H5, H6 and H7. If we see extracellularly, that is outside the cytosol, 3 loops are present, E2, E3 and E4. And if we see intracellularly, there are 3 loops present, C1, C2 and C3. And there are free ends where one end is attached with amino group, the other end is connected with carboxyl group. And if we see this GPCR are having 3 families. One is A family, that is rhodopsin family. So the example of these types of receptors are amine neurotransmitters, purines and cannabinoids. And B type of GPCR receptors, which are also called glucagon receptor family. The examples include peptide hormones. And C type of receptors, which are also called metabotropic glutamate receptors. The examples include GABA B as well as glutamate receptors. And if we see here the pictorial representation, so here it is having 7 helical structures and here it is having alpha, beta and gamma. So when this G protein, it binds to the subunit, then the GTP is converted to GDP and thereby it is altering the mechanism. So here the GDP is converted to GTP and this is a cyclic process. So GDP and GTP conversion takes place whereby the ATP, that is adinylyl triphosphate, is converted to cyclic AMP. First, here this initial stage is important in order for it to exhibit various pathways. So if we see the subtypes of G protein receptors, so this G protein receptors are having various subtypes. GS type of GPCR receptors which are present in beta adrenergic, which are examples include beta adrenergic amines, glucagon, histamine and serotonin receptors. So here these receptors, they undergo the mechanism of cyclic AMP pathway showing excitatory effects. And GI type of receptors, examples include alpha 2 adrenergic receptors, muscarinic acetylcholine receptors, opioid and few serotonin receptors, which are exhibiting the pathway of decreased cyclic AMP levels, thereby showing inhibitory effects. And GO type of receptors which are present in the olfactory epithelium and they undergo increased adinylyl cyclic AMP pathway. The other type of receptors are the NT, that is NT type of receptors, opioid type of receptors and cannabinoid type of receptors, where the signaling pathway exactly is not known. And if we see GQ type, which are present in muscarinic acetylcholine receptors, serotonin receptors, especially 5-HT1C type of receptors, where they follow the IP3 and DAC pathway. And GT type of receptors are present in rhodopsin and cone cells of eye, where they undergo the process of increased cyclic GMP levels. So here in this GPCR, as I told you, they are having various signaling pathways. So this GPCR, they are having the 4 different types of pathways. One is cyclic AMP pathway, inositol triphosphate pathway, ion channel regulation and rho kinase pathway. So if we see cyclic AMP pathway, the GPCR, when it is getting opened, the drug molecule is entering, and the GDP, it is binding to the alpha subunit, and thereby the GTP is converted to GDP. So this conversion, it will help the ATP to convert to cyclic AMP with the help of an enzyme called adenylyl cyclase. So if the cyclic AMP levels are increased, then it shows excitatory effect, and when the cyclic AMP levels are decreased, it shows inhibitory effect. If you see the IP3 pathway, here the mechanism, initial step is same. GTP converted to GDP. After whatever the process is there, that is different. So here once the GTP is converted to GDP, here with the help of, here we can see PIP2, that is phosphatidylinyl diphosphate and diacylglycerol. So this PIP2 is converted to inositol triphosphate, and this inositol will aid in the calcium levels. Intracellular calcium levels will be increased with the help of this IP3. So if intracellular calcium levels are increased, it shows excitatory effect, and if calcium levels are decreased, it shows inhibitory effect. The other one is Rho kinase pathway. So Rho, it is an important factor here. So the GDP, once it is converted to GDP, so the Rho, it is converted to Rho cyclic kinase. So when Rho kinases are formed, so these Rho kinases, they play a very important role in the conversion of myosin light chain to myosin light chain phosphate. So they will allow the phosphorylation of myosin light chain with the help of an enzyme called myosin light chain kinase, and thereby they are aiding in the contraction of the muscle fibers. The other type of receptors are enzyme linked receptors. So here these enzyme linked receptors, they are involved in various types of mechanisms like growth. They are helping in the growth, they are helping in the proliferation, that is cell division, and differentiation. And there are various chemical mediators, or there are various enzyme mediators which are there, like cytokinins, hormones, insulin and leptin. So all these mediate the action of these receptors. Here these type of receptors, or the mechanism by which these types of receptors are working, is very slow, because the drug has to bind to the receptor, gene expression has to be changed, and then only the receptor shows its action. And if we see the types of these receptors, there are receptor tyrosine kinases, which the examples include EGF and NGF and insulin receptors, and serotonin or threokinase receptors, that is TGF receptors, and cytokinin receptors like cytokinins and CSF, cerebrospinal fluid receptors, guanylyl cyclase receptors that include atrial natriuretic peptide, that is ANP. So if we see the MAPK pathway, so here this enzyme link pathway, it has two mechanisms. One is the MAPK pathway and the other one is JAK-STAT pathway. So this MAPK pathway, it is activated by a mediator called tyrosine kinases. So here if we see EGF is nothing but epidermal growth factor. So this pathway is aiding in the growth. So this epidermal growth factor, it binds to the epidermal growth factor receptor, and thereby it is potentiating RAS to release, RAS. It is a chemical mediator, and this RAS, it is converting the GTP to GDP, and this will allow the MEK to release. So if MEK is released, then this MEK is converted to MAPK. MAPK is nothing but myosin activated protein kinases. So if these kinases are activated, these kinases, they aid in the proliferation or cell division by changing the gene expression. So this pathway is mainly important in cell division, growth and differentiation. The other one is JAK-STAT pathway. So JAK-STAT, JAK in the sense Janus kinases. So here this cytokine, these Janus kinases are various, like they include cytokinins, interferons, interleukins, like chemical substances in the body. So this cytokinin, when it binds to the cytokinin receptors, so the JAKs are released. interferons, interleukins are released. So these, what do? They give some space for the stat-like compounds to bind to it. Stats are nothing but the protein substances. So here they are giving some space for the stat-like compounds to bind to it. So stat-like compounds are nothing but which will alter the gene transcription. So here if this space is provided, these molecules will come and bind at that space, and thereby the transcription mechanism of gene expression is altered. If we see the last type of receptors, which are called nuclear type of receptors. So these nuclear type of receptors, they alter the gene expression of the cell. So this ligand, when it is having a space for ligand. So when this ligand binds to these receptors, they will alter the gene expression. And this is present in soluble form. So these receptors are present in the soluble form, either in the cytoplasm or in the nucleus, and they aid in the signaling pathways. They will transduce the signals by modifying the gene transcription, and they play a vital role in endocrine signaling, because this endocrine signaling, the hormones are the one which where the hormone levels are to be maintained. So these hormones, they follow or they bind to these nuclear receptors only. If we see the examples of these receptors, steroid hormones, glucocorticoids, vitamin D and vitamin A receptors, and thyroxine. So these all are the examples of nuclear receptors. So if we see the structural organization, so how the structure of these nuclear compounds look. So it has an N-terminal domain where the amino acid binds, and it has a DNA binding domain where the gene transcription takes place. And it is having a ligand binding site where the ligand bind to it, and it is having finally the C-terminal domain where the carboxylic acid is binding. If we see the types of these nuclear receptors, there are three classes of nuclear receptors: class 1, class 2 and hybrid class, which is a combination of these two. So class 1, they are present mainly in the cytoplasm, and hybrid class are the one which are mainly present in the endocrine system, and class 2 are the one which are present in the nucleus. And if we see class 1, they are forming homodimers. Homodimers in the sense they are forming single compounds. The two molecules combine, they form dimer. So one dimer is formed in these type of receptors. And if we see hybrid class, they are forming heterodimers. Different dimers are formed. In class 2, here also they form different dimers, but here the lipids are the one which are the mediators. And if we see, the class 1 is having high affinity binding, and class 2 is having low affinity binding. So if we see the class 1 type, here we can see the hormone, it is binding to the ligand binding site. Here LBD is nothing but ligand binding site, and thereby it is forming a complex with the ligand binding site, and this complex is called dimer. So this hormone, when it is forming a complex with the ligand binding site, it is called dimer, and this dimer will alter the gene expression. So it will modify or it will alter the gene expression mechanism. And here if we see this, here class 2 type of receptors which are present in the nucleus, so they bind to some mediators called RXR. So when these bind, they form different dimers, and thereby they are aiding in the gene expression. So if we compare the receptor types, so inotropic has 483 families, metabotropic, that is GPCR, have 217, kinase linked have 135, and nuclear receptors have 48. And these ion channels are present on the membrane. GPCR and kinase also are present on the membrane, whereas nucleus is present intracellularly. And if we see the time taken, so these ion channels, they show the action within fraction of seconds, and metabotropic show within seconds, and kinase and nuclear should take little hours, few hours to show their mechanism. And coupling, we can see the drug molecule directly binds to the receptor in ion channel, kinase and nuclear, whereas in GPCR it is the mechanism is showing through G protein type. And the examples include the ion channels have nicotinic acetylcholinergic receptors and GABA A. GPCR include adrenoreceptors and muscarinic cholinergic receptors. And kinase linked examples are insulin, cytokinins, and nuclear type of receptors are examples of steroid receptors.